Professor and member of Research and Innovation Council and Chairman in Institutional Ethics Committee. Is Adjunct Professor at Asia Pacific Institute of Embryology, Mysore. Then Consultant for Human Genetics Unit, All India Institute of Speech and Hearing. Adjunct Faculty for Biotechnology, Mangalore University. And Chairman for Institutional Ethics Committee, Mysore Medical College and Research Institute, Mysore. He is coordinator for MOOC courses on human genetics and principles of genetics in CEC, UGC, Ministry of Education, New Delhi. And he has vast past experiences. He has worked as UGC-BSR faculty fellow at Department of Studies in Genetics and Genomics. And he has worked as professor and founder chairman of Department of Studies in Genetics and Genomics. And he was professor and coordinator for MSc in Genetics in the Department of Studies in Zoology. And he has worked as visiting professor for one of the university in Austria. And he has worked as senior research associate for University of California, Los Angeles, USA. And he was reader in zoology, Mysore University, lecturer in zoology in Mysore University. And he has worked as postdoctoral scientist at McMaster University, Canada. Is an alma mater of Mysore University with PhD and MSc also from Mysore University. He has guided more than 30 completed and still 2 are working under his guidance for PhD. He has attended more than 320 national and international conferences, and his publications number up to 285. And he was in different administrative and professional responsibilities. It was more than 110. And he has handled research projects and successfully completed 18, which is from DBT, DST, CSIR, ICMR, UGC. And he is also a life member of many national and international professional societies. To his credits, he has 25 H index and 69 I-10 index, and total citation to marks up to 3,000 plus. So it is a remarkable achievement on Professor Ramachandra sir. And for all these achievements, he has been given a lot of recognition and awards. In 2023, he was elected fellow of Indian Academy of Biomedical Sciences. In 2021, he was honorary fellow of Karnataka Science and Technology Academy. In 2021, he was awarded Shishyaru Mechida Guru Award from Negile Yogi Samaja Seva Trust. And he was a NAC review expert member. He is also FASC elected fellow of Indian Academy of Sciences, Bangalore. And he has won University of Mysore Golden Jubilee Foundation Award for the year 2014-15. And he has worked as local secretary in the ISCA, which was held in 2016. And he is Dr. Raja Ramanna Scientist State Award in the year 2014. And also he has taken Professor G.K. Manna Memorial Award in 2014-15 in the ISCA held in Mumbai in the year 15. And is also a recipient of C.V. Raman Young Scientist Award for Life Science. So the CV goes very vast. And with this brief introduction, I welcome you, sir, to this session, sir, to this afternoon session of Know Your Genome. Thank you very much.
Thank you, Dr. Hemalata.
This is Rajkumar, sir.
Yeah.
On behalf of all the participants and on behalf of University of Mysore as well as UGC-HRDC Center, I welcome you, warm welcome you, sir. Yeah. So I request you to present your talk, sir.
Yeah. I at the outset, I'll thank Dr. Hemalata reading my biodata and inviting me, Dr. Rajkumar, for giving this talk. So I'm very much delighted to share my understanding, reading, and my own work with regarding to genome and genomics. Dear learners and participants, you can stop me at any time and we will have a discussion, introductory sessions. So I don't hesitate any time to stop me also this. Yeah, can you see my slide here? Is it my slide visible?
Yes, sir. Yes, sir. Yes, sir. I think you should put it in slideshow mode, sir.
Yes.
Yes, sir. Slideshow mode is required.
Yes, I'm keeping it. Is it okay now?
It's no, not yet. Not yet.
It's a slide show. I have made it a slide show. Okay.
In your PPT, I think you should open in slideshow mode, sir.
Okay, then I'll go back. Yeah,
Yeah.
it opened. I'll go back. Chart. Okay. Yes. Close this. No,
Well, if you press F5, I think it will come. F5.
yeah, yeah.
Solutions.
Can you see now?
No, sir, not yet.
So I put it in a slide mode.
I think you should do share screen now, sir, after
Oh,
the slide mode.
Okay, then I'll go. Yes. Yeah, I'll come here. Yeah, this is my share.
Yes, sir, it's correct.
Is correct now? Yeah.
Yes, sir, we can view.
Yeah, okay. Yeah, friends, the topic I chose is the, let me put them in, yes, yeah. Know your genome, and my outline of my talk runs like this. I'm going to briefly tell about, I hope all of your life science background, most
Yes,
of the
sir.
things you are already aware. I wanted to compile to make a story of that, how far we are learned or we know our own genome and where we are going in future. Now, whether we like it or not, and we have to understand our own genome for our better health and so on. So in this direction, I was fascinated from 1985, reading when the Human Genome Project was conceived and as a pilot experiment. From then onwards, I'm a follower of almost all the article, whatever I could able to reach. I'm happy. So this is the information what I learned as well as my work in my lab, what we have done here. I want each one of you to read this the phrase. And what will be the answer?
Because of genes.
Any others? Any other answer you have? Well, let us not wait. Yes, she is right. Genes, earlier used to call genes or genetics, no, genomes. Genes and genomes are the one responsible for what we look today. And like we look all the persons who are born to the human or look like the human one. What are genes? Gene is a basic unit of heredity which determines the nature and function of the cell, and it controls the phenotype and by specifically synthesizing the polypeptides. Gene encodes protein required for the growth and maintenance of any cell or an organism. Johnson coined the term 1909, the gene, that time gene. Can you see this image? Can you test in your with you
Yes,
or your family and your friends?
Yes, sir.
these are the human traits. I don't know my slides, the title of my slide is not coming. It's anyway, I can manage this. All these are the traits, and each one of them are, controlled by a gene, and all the traits, whatever we have, are controlled by the genes. We inherit our traits shared with grandparents, shared with parents, shared with brother or sister, the siblings, and also we have our own unique kind of trait ourselves. How do you say that we have a unique traits? What could be the reason? Any idea? Because of meiotic recombinations, and every sperm and egg what we have developed when we are born is a unique gametes, and they have a unique recombination. That's why we do have our own unique traits. What is a genome? Is nothing but a total DNA of an organism, including genes and non-coding sequences. Genome is encoded either in DNA, majority of the organisms, but in some viruses, as you know, the RNA is the genetic material. If you just look into the size range of the genome of variety of organisms, including plants, animals, the protozoans, those simple, the single cell organism, the vast majority, the size of the genome is varying. I hope you can see here, 15 MB to 150,000 MB in protozoans. As the organisms or the life evolved in animals, look at the birds and the mammals, the narrow range. Okay, around 3 billion base pair, 3 to 4 billion base pairs. See, all the mammals are there. Let it be elephant or a mouse or the human, the range is less. But if you look at the flowering plants, also vary in a genome size. What could be the reasons of varying a genome size in flowering plants? Any idea?
Large number of proteins present in them.
Any other answer?
Mutation, sir.
Yeah?
Evolutionary origin, their origin evolutionary mechanisms, maybe. They have evolved and originated and evolved in different areas of the world.
Right, they are tetraploid, polyploid plants. That's another. And the repeated DNA is much in the higher organisms. Okay? I hope you know that many of the plants are tetraploid and polyploids. That is the reason. Okay? Why should we study the genome? I'm audible, right? Am I audible?
Yes, sir. Yes, sir.
Yes, sir.
And
You are
our
audible,
slides
sir.
are visible?
Yes, sir.
Okay.
Yes, sir.
Okay, thank you. If there is any problem, please let me know, because I'm not in virtually in front of you, but I'm on online. Okay. Each and every one is a unique creations and life little book of instruction, that is our ATGC, and a hidden code determines which protein should be made and when, and the language, that is ATGC language, nucleotide language and amino acid language, is common for all organisms. Look at all the organisms, including prokaryotes and eukaryotes. Any change in the genome sequence, I hope you know the total DNA coding and non-coding is the genome of any organisms, and any change in the genome sequence leads to abnormality. We call this as an anomaly or the disease. And more than nearly 6% of the humanity is with genetic diseases. That means around 6 individuals out of 100 are suffering from genetic diseases. With the advent of our technology, from the recombinant technology from the 80s and later genomics, the scientists are asking questions like, can we do something for this unrepairable, irreversible genetic diseases where our kith and kin are suffering with the diseases with this technology? That was the initiation of the Human Genome Project. And it's a long story, but to make it, the Human Genome Project has conceived in 1985 and finally started 1990, completed 2003, and they planned it for 15 years, but it has they completed it with the advance of technology in 13 years, but including the 18 countries, started with the US Department of Energy and NIH, and the other 18 countries are involved for that. What are the objectives of the one? Identify all the genes, determine the sequence, store the information in databases, develop the tools for data analysis, and address the ethical, legal, social issues. So this has been compiled. To make the story very short, in 2001, February, Bill Clinton was then the American president. He made the private funded as well public funded to come together and compile the working draft of the Human Genome Project, and they published in the public sponsored one, published in Nature, the working draft, and Science by the Silara Genomics is a private funded one. And completion of the sequence of Human Genome Project was announced April 14, 2003, to commemorate 50 years of double helical structure. I believe each one of you aware that the Watson and Crick, they demonstrated the double helical structure in 1953. So now, what is genomics? Genomics is nothing but the study of genomes by using a set of techniques, analytical methods, more than that, asking right kind of scientific questions to complete the genome. So this has started in 1985 with a smaller genome, and later the human genome 1990 onwards. The methodology used was, hello? Any doubt? Anybody has question there? So there was some disturbance? No?
No, so you can continue some disturbance.
It has started to plan very well. They planned for 5 years, 3 phases of the project. Well, then we call as step by step. First, they looked into the genetic maps. Then they created the physical map. Then each piece of the DNA, they sequence and align them together. It is a very laborious work. To compile the whole the 13 years work, the genetic components in the genome is 1.5% is the coding sequence, protein coding sequence. About 26% is the introns, and about 20% of the line sequences. Lines are nothing but long interspersed nucleotide elements. About 13% of the short interspersed nucleotide elements. With the long lines were around 6.5 kb each one, and whereas sign was about 300 nucleotide each one, and lot of other repeated DNA, transposon, dead transposon, or many other segmental the repeated sequences we do see in our genome. That means the 1.5% is the coding sequence. The rest of them are, majority of them are repetitive DNA sequences. We have a 3.2 and an average base pairs of our genome and about 25,000 protein coding genes in our system. How much we are similar each other between us here? 99.6% an individual identity has been established with the Human Genome Project as well as later genome analysis. But how much we are different from each other is 0.4% equals to 4 million base pairs we are different. That's why we look different. So genes and the size of genome in each chromosome has been very well established with our genome project. The first chromosome having about 3,000 gene and 279 MB, and the ninth chromosomes is about 1,299 genes and 140 MB. And whereas if you compare the chromosome 9, where I have marked here, chromosome 11 chromosomes supposed to be smaller than the 9th chromosome with a conventional or banding method. But however, when you analyze the genome, 11th chromosome is 148 MB, whereas 9th chromosome is 140, whereas the 11th chromosome has more genes compared to the 9th chromosome or the other chromosomes. Like that, there are many comparisons can be made. You know, here the 19th chromosome having double the genes as the chromosome 18. 18 chromosome is having around 700 genes, whereas 19th chromosome has 1,454 genes. And they have culprit chromosome for Down syndrome having about 303 genes, whereas X chromosome, where the female have 2 X chromosomes, have around 1,184, around 84 genes, whereas Y chromosome has very few genes, that is the smallest, the lowest gene number found in the Y chromosome, that is through 31 genes. They are mainly involved in the fertility as well as the maleness gene, involved in the whole. Also, for some of them are for the growth and development of an organs. What is the size of the gene? If the genome project, I'm just taking some of the novel significant contribution or the discoveries. That is, the range of the gene size varies from in eukaryotic system from 21 or human but 21 BP base pair to 2,400 BP. That is 21 base pairs called as microRNA. It's a non-coding genes. We have around 6,000 microRNA in our system. Every one of us has that. They are very, very important for our growth and development of or maintaining our even they involved in proper gene regulations. And the smallest protein coding gene is the insulin-like growth factor, is having 252 gene, 252 base pairs, and 60 codes for 67 amino acid. The longest human gene, I hope most of you know, that is dystrophin, present on X chromosome, having about 78 introns and 79 exons, about 3,685 amino acids. This dystrophin gene is equal to half of the bacterial Escherichia coli genome. Bacteria has about 4 MB sequences, whereas the gene itself, one single gene itself, 2.4 MB sequences, that is 2,400 KB. The human has very typical the antibodies, specific antibodies, T cell receptors for antigens, antigen for the major histocompatibility, cell signaling molecules, cytokines, and blood clotting factors and apoptosis factors are very unique to human. They are present in our body, in our system. With this, the summary of the Human Genome Project, when the Human Genome Project was announced 2003, even 2000 itself, people have started asking question, is it going to be sufficient for us? Is it going to represent all the humanity? several questions are being asked, and then they started a newer genome project. That is one of the genome projects. Several of them started. The first one they started was the international HABMA project, about 270 individuals of 5 different origins. They sequenced that they wanted to identify the common variants found in different the ethnic groups. And later, the company started individual genome project like Whalers, the color 3. The company sequenced the Watson genome for $2 billion. They spent $2 million for that. And 2007, Craig Venter, phylogenomic the head, he sequenced his own genome 2007, and later individual genome sequencing was also possible by now. Then 2008 they started 1000 genome project to understand more, ENCODE genome project, synthetic genome project, personal genomics, 1 lakh genome project, several 1 lakh genome project, UK project and Asian project have started, genome editing concept, gut microbiome sequencing, and pan-genomics. From last few years they started finding out different ethnic groups and the genome, commonality of the genome. And now last week he had announced that the Indian government at the 28th of Science Day announced the Human Genome India project where 10,000 genome normal individual genome sequence has been sequenced. I'll come to that point later. So several genome project has started after the Human Genome Project, understanding more about that, understanding the broader application of that, and also how much that is helpful for the whole humanity. They started new project. This has generated a lot of economy in the American government or even Chinese government and UK government, because this has helped to utilize the machinery and so on, even the developed machinery to export the machinery for other country wherever they use this. Now, I wanted to say these two individual. I think this individual, yeah, I think you can see his head right here. Sultan and Pink. Can you see these two individuals? And he has 8 feet, and this Pink has only 2 feet. Are there diseased persons?
Yes, sir.
How many of you say no or yes?
Yes, sir. It is
No, sir.
Yes, sir.
So we have different opinion, right? Yes and no. I don't think, I think I have my slide, I don't know, this one. They are not diseased persons. They are very in size. They eat the normal food like anyone. And that is because of the genomic variation. The height is one of the trait. What we can see is the height here. The height is controlled by several genes. And we do see a genomic variation of the height controlling genes. That's why they do have. Now we have the scientists to identify which are the genes involved in the polygenic trait, the height. And this is because of genomic variation, not because of the disease. So the changes in the genome sequence of different individuals, we can see there. So now, the question is that, what are the genomic variations? Who is normal? Who is normal? Are we a normal? Or how do you quantify the normal? Normal human being. Or in animals or plants, we can say it's a wild type animal, wild type Drosophila fly, and so on. So we define the normal human based on the most frequent in the population. By looking into the nucleotide bases, and 99.6%, and we are identical with the genome sequences. That's why in those nucleotide we are normal, but we are different by 0.4% of the nucleotide sequences. So There are two terminology one has to know when you understand the genome variations, that is polymorphism. The DNA polymorphism we call single nucleotide polymorphism. These are the one more frequently seen in our genomic variation and in our genome. They are 1%. Out of 100, they are around 1% of the variations we see. They are called a polymorphism. They may not always they cause the disease, but however, now scientists are targeting the unique variations found in our genome, in the gene, and there can be causative factors for the genetic diseases. The most common term, I hope all of you know, that mutation. What is mutation? Many of you may say that sudden change in our genetic system. It's not so, it's not a sudden change. It's a heritable change. It's a very, very rare event. 1 in 1 million to 10 millions, which has been quantified in bacteria, they are the mutation. They are a spontaneous mutation. They are very, very rare. Generally, when the mutation occurs, the people used to call it is causing disease-causing mutations. In the beginning, in 1900 middle, when the people, the scientists have identified the mutations by using chemical agents, physical agents, so on. But it is not always so. So there are genomic variation increased by mutations, sexual reproductions through meiosis as well as fertilization. Both the time they undergo the recombination there, and also the terminology what I used here is inbreeding. How many castes are there in India? We have more than, I don't have time here, so I'll tell the answer. More than 6,000 castes we have. It is more than 4,000 castes, 4,600 and that. So each caste invariably is inbreeding one, but overall, if you look at the Indian population, we have diverse, but however, we are inbred populations. We are unique to the whole world in our many features. This one is our genomic variations, right? So the inbreed populations, and we need not have the similar kind of sequences, similar kind of genomic variations, what we found in Western data, and we need to establish our own. This is the questions we have to ask, and this is being done from last 20 years from several labs in India, and we did achieve a lot in that. I'll come to that point later. So non-lethal mutations are the reservoirs of the genomic variations. So what are these non-lethal variations? Any recessive gene mutations we harbor because they need 2 copies to be expressed there. So they are not lethal. If it is lethal, embryonically, the embryo will not be developed, they are lethal. So the mutation which are there in our system, they are recessive mutation we harbor. We are the storehouse of the recessive genes, the mutated recessive genes. So we now know that chromosomal aberration like deletion, duplications, inversions, and translocations, which have been taught from high school to the even degree, it is there in textbook, but now the genomic variation, we need to know, we need to tell, we need to establish this or to teach our students. That is, the 4 major genomic variations are SMP, single nucleotide polymorphism, microsatellites, few nucleotides are repeated randomly or deleted in our genome, and indels means insertion and deletions, around 100 nucleotides are added and deleted in our genome. The 4th category is a major category, is called as copy number variation, that is the CNV. We do have from 1 KB to 5 MB sequences in our genome is deleted or added, duplicated. This causes major problems, major diseases in our system. So I will not go into more about that. From 2012 to now, we have started last 10 to 12 years, we started working on the CNV in our lab, and we produced many the ask scientific questions. We are pioneer in that in my lab, and that has been recognized. My contribution CNV and the recognition, whatever the faculty, the fellow of Indian Academy of Sciences, is because of my work on the CNV and other contribution and so on. So I'll go very fast. The copy number the single nucleotide polymorphism, the transversions. You know the meaning of this, and they can go synonymous on synonymous variations, and we know that one nucleotide change in a sickle cell anemia change the amino acid, even the lamina or the oval shape of the blood cells become sickle. It affects the carrying of the oxygen there. And also trinucleotide, if the trinucleotide repeats are more than the required, more than the normal one. For example, here, look at the Huntington disease. If they can CAG repeats can be up to 35. If it is more than 37 to 100, then it end up with the Huntington disease. Even like Fragile X syndrome, that CGG up to 54, it can be tolerable. If it's more than 200, it's a mentally retarded individuals can be seen there. So trinucleotide repeat may either in the coding or non-coding regions. There are several examples are there. Insertion and deletion can cause varying the genetic code here, coding sequence genetic code. They can create a frame shift mutations. They can present randomly anywhere in the genome, anywhere in the gene have the coding sequences and the regulatory sequences and the flanking sequences. Anywhere this frame shift mutation or the insertion deletion can occur, they cause the mutation. So we did a lot of work on the SMP and the indels. We did some of my PhD students are taken as the problem of this. In diseases, this congenital heart disease, asthma, and even in a normal cohort of debility population, we could able to make some story out of it. The fourth category is the copy number variation. Is, as I told you, it includes the deletion, insertion, and duplications. It is more deleterious. For example, now, what are the causes for copy number variation? Copy number variation, I say that because we are a diploid organism, we have two copies here. Because of homologous repeats, segmented duplication, chromosomal rearrangement, duplicative transposition or non-allelic recombination, we can have one copy and none of the copy, or 3 copy or 4 copies. So this increasing copy number called duplication, decreasing copy number called deletions. This is going to be effect for any individual. They may cause the disease because larger chunk of the sequence up to 5 MB, many genes might be involved in the 5 MB regions. They are deleted or duplicated. They become inactivated. That's what it causes. So some of our own work, what we did on understanding of the CNV, to briefly to explain one of the example, we will take that we studied the 1,715 individuals belong to the world 12 populations. These are the different populations. Our own Indian population of 43, where we worked we studied the 2.8 million markers on these genomes, and we studied of this using this. We got these genome sequences, raw data of the unprocessed data from affirmative genome-wide SMP 6.2 array from UBI, European Bioinformatics Institute, by MOU. Only research purpose we used, and we used many algorithms, many softwares to analyze the CNV, and we quantified the data CNV of this 1,715 individuals. About 75% is the duplication of the copy number variation seen in all the 12 population, and 25% is the deletion in the genome. If it duplicated, organism can survive. If it deleted, that whole protein is been lost there, then it become deleterious, it causes the disease. The smaller 1 to 250 kb size variations, copy number variation, we did see they are more frequent compared to other copy number variation. We mapped all these copy number variations, about 44,109 copy number variations for all the 24 human chromosome, and an average 1,329 genes per genome have the copy number variations. And also we mapped in the coding and non-coding regions of the duplication and deletions. We do see here the dark green is the one is called as a duplication. We can see that duplication African population, European population, as well as the Jewish populations. Even the India has a more duplicated segment, India, Australia, and Taiwan populations here. Whereas here, look at the China, here the deletions are more in China compared to the duplication here. So we mapped even the deletions, we do map them as a different population. We summarize them, the important role in cellular processes, and also we use copy number variation as a genetic marker to trace the human migrations, and which obeys with the already established that the out of Africa human arise from the Africa, and the second settlement, we established the second settlement in Asia, and from there they dispersed to the New World, and also based on the genomic, the copy number variation found in the Africa, less 3% only genomic variation, whereas we found in America 16% genomic copy number variation, and we shared this also. This we have published in PLAS one, very well accepted, and the New World has highest number of copy number variation. Even many the latest the people who works on the human evolution migration refer our article in this. And also we use the copy number variation to identify the type 2 diabetes mellitus risk genes. And one of my MSc project students, Manasa, she worked on that. We this copy number variations in the genome as a project student, and also we made a publication. I'm going to tell the story very shortly. If you need, you can refer our article, you can write to my email, or you can ask me later on the questions. Another very interesting message in the copy number variation, we found the dyslexia, where the reading disability we see in the school going kids. They have 10% of the school going kids or the dropouts from the schools, they are dyslexics. So we have to take care of them very well. We have to diagnose them early stages. We have to give special training, and then only, I hope you have seen the Tare Zameen Par movie from the Amir Khan there, elegantly use this concept there. So we found the protocadherin gene, which is present in X and Y chromosome, is defective. It is needed to hold the two neurons together. The cadherin, that is protocadherin, is needed. And we did a lot of work on that. We found regions within the X chromosome and the Y chromosome, P regions. There is a new recombination between these two. In this family, worked on that. That was a generally human X and Y chromosome do not undergo recombination except the terminal regions here. I think I'll go to the next slide. This is the paper we published. I'll come back to the one here. Here, PAR 1 and PAR 2. PAR 1 means pseudo autosomal region 1 and 2. They do undergo similarity. They have similarities between X and Y chromosome, undergo recombination very rarely. But we found, because of dyslexic, the patients, the kid have the Y chromosome gene has been transferred to the X chromosome. We identified this as a Part 3, new recombination region, which is new to the literature. This is our new discovery. This has been recognized by academy, and also now I have good many citation, because part 1 and part 2 have been discovered 1980s between human X and Y chromosome, and this is the new discovery. People from different part of the world, they appreciate the new region. They also tested and they qualified that our discovery is right. And we made a lot of the analysis asking different kinds of diseases, and we made the publications here. Okay. Another genome, our own genome, human genome has been sequenced. One of the patient here, we found the June 29th, 2013, in Times of India, one of the patient has these features, like is a made 35 years old, it has a very clean family history, doesn't have any of the family members have the cardiac arrest or the atherosclerosis, but he has the condition of myocardial infarction, transient ischemic attack, that mild stroke, and type 2 diabetes. All these are, but his lifestyles are no smoking, non-alcoholic, no high cholesterol or poor style, but however, his arteries are blocked. Like this arteries, wherever the arteries are 80%, 95%, they are blocked. And using American online, the angioplasty they did it, but still it was appearing that. And these doctors from this Gujarat, they are prescribing the drug generally by looking into this, the condition of these kind of drugs they're prescribing. But we finally found this patients MOU. We established MOU with the doctors, and we got their sequence. We got their blood sample and they sequenced them. We analyzed the transcriptome of them. To make the story short here, 13 the strong mutation we found there, out of them 11 are non-synonymous, 2 of them insertion. All this mutation was seen in exons. There are 4 major genes have hit there. They are involved in the processing of the cholesterol. So the blockage of the cholesterol accumulating in the arteries, and we do see all the importance of the gene. Based on this, our genetic data, our genomic data, doctor prescribe based on the defective gene as a different drug, which can help to overcome and not to reduce the blockage of the arteries. It was successful, and they say even for 2 years, the blockage was not that extent what earlier they had. So this is like knowing the genome sequences, understanding the genes involved in that, the protein involved that, and describing the individual specific drug is called genomic medicines. Okay, now how our genomes have been evolved. I think how much time I have? Another 30 minutes. We can also have a discussion later. So how genomes have been evolved? Do you have any questions so far? What I have covered, genomic variation, genome projects, and broadly about the genomic genome. Do you have any questions now? Or any clarification if you want? No?
No, sir.
Okay, but I do not, I presume that all of them are listening to my talk, right?
Yes, sir.
Okay.
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay, thank you. So now, see, we have a 0.4% genomic variations. One of the variation, several variation, one of variation, what we analyze the global genome, I just gave some of our the reports, what we have done, the investigation, what you have done, and the patient with this, so the very specific the disease, the heart disease, atherosclerosis, that we analyze them, also genomic variation found. So there's a lot of evidences that genomic variations, if you identify, we can treat them, manage the patient also. That is the kind of the message I wanted to complete there. I'll come to the next point there. How genomes have been evolved there. So only 1.5% of the coding sequence I told you, majority of the reproduced DNA sequence, that is because of duplication. I told you copy number variation is more in our genome, and the rearrangements, mutation of the genome contributes to the genome evolutions. So from the prokaryotes to the first eukaryotes, there were generally, if you look at the bacteria, is around 4,000 to 5,000 different bacteria have the genes. And the first transition from the prokaryotes to the eukaryotes about 4.4 billion years ago, it's about from duplication of the gene, that is about 10,000 genes. We have a lot of evidences for that. And the second transitions for the evolution of evolution of genomes in organisms, eukaryotic organisms, is that first vertebrate at the end of the Cambrian, that is 530 million years ago, from 10,000 to the 30,000, where all the mammals have been evolved there. So there is two transitions from prokaryote to the first vertebrate, from first vertebrate to the second transition to vertebrate to the mammals there. So there's two transitions. How the new genes have been acquired, there are two ways: by duplication of the existing genes. Now we know multi-gene families, globin multi-gene families. They are very, very essential. RNA genes are the thousands of copies of RNA genes are essential. They are there, and even the duplicated gene acquiring the genes from other organism. We know that the basic the lateral gene transfer phenomena in bacteria, transformations, conjugations, and transductions, they are very, very rare, but we do see laterally genes have been percolated into the organisms. And even the lateral transfer between mitochondria to the nuclear genome to the chloroplast genome to the, I'm talking about overall the eukaryotic system, not the chloroplast. We don't have a chloroplast. Don't think that I'm telling that chloroplast is present in the human here. So to make the genome, eukaryotic genome evolution, there is all level of the inter-exchange of the genome. There are several evidences are there. The same copy is present in nucleus, or chloroplast, or mitochondria, or vice versa. So our genomes have been evolved. We do see that there are two phenomena. One is lateral gene transfer, another gene duplication, acquiring. They already do. We do have multiple genes, polygenes, as well as the multi-genes of the one family, as well as the same gene can acquire a new function there. So now let's review some of the breakthroughs, how one can appreciate the genomic studies there. Many of the even teachers or the humanity are always greedy, that tell that I'm not happy, I should have got more salary, I should have become the director, I should have become vice chancellor, I should have become governor. Always the greedy, they are not happy whatever they are earning there, even the salary, whatever getting and there. So they become always, anything you say, they think that I'm right, others are wrong. They do have problems of regulating the happiness genes, that is serotonin. Serotonin in our system has to be regulated properly, then only we'll have satisfaction. At the end of the day, we have to have satisfaction. If we don't have satisfaction, then we'll not have a peace of mind there. So scientists have identified the serotonin regulatory moiety, and if somebody has depression, somebody has this kind of problem, sometimes they will admit to the hospital. Indirectly, the doctors give derivative of the serotonin regulatory, the enzymes or the activators there. So The secret is the gene that releases the happy chemicals into the brain that makes us too happy there. So please let us not be greedy. Let us always try to have satisfaction with that. Then we can live a little more longer and happier and healthier there. We do have a sports genes. All of them are why Chinese or even Africans are winning the Olympic gold medals, because they do have a very, very fast regulating muscle genes. And fast muscle fiber, that is ACTN3, has to be normal and regulate the function. If this is having any kind of variation, mutation, then even whatever the way kind of training we go, it's not possible. It is being proved. Now people, the scientists have taken from the sports school to identify these genes, to see that can we train this person for the competition, for the Olympic and Asian Games and so on there. So I had, I don't have time now. I had an interesting case. Some parent has called me that you are a genetist. Can you identify the gene? My son has a gene for going for the Olympics, but I am preparing. I asked him, how old your son? Are 7 years. And he told that he is spending every month 50,000 rupees for good nutrition, but I wanted to know my son has a good gene and so on. So finally, we did the exome sequencing of those individuals, and we found that gene is normal in that case. But later, we are not able to trace them. They are not available. But they are from Mysore only. So the loss of ACTN3 is deleterious muscle production, or even variations there. It's a deleterious. Third example, what I wanted to give here is the color blindness. Many of us, about 9% of the whole humanity, is colorblind. So colorblind, they are not able to recognize the red light and the green light, and so remember in the signal light. Many times they go with the flow and they see the either side, then they move. So they have a defect. They cannot identify red or green. It's a gray. Sometimes they end up with the accidents there. So, friends, this 9% means out of 100 members, there 9 members have this in the whole the world, the human population. It's a very, 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 very important. And now we do have corrective glasses. So just they need not reveal to others. They can go to the doctor, tell that I have this one. They can give a corrective glasses that will rectify to identify green or the red. And this is available, but many people ignore that. Then we'll have a lot of problems in the signal regions there. Whatever the medicines we take today, all of us, even including doctors, is a trial and error medicine. They think that based on the experience, based on the situation, oh, this bacteria is inspected, you have a cold or cough or fever, they will give. Always they may not be successful. Then they, after 5 days, will go to the doctor again. That sir is not, doctor is not, the fever is not coming down, or my cold is not gone. Then he will prescribe another, and he will prescribe another 5. He will prescribe. Sometimes we do have resistance to this kind of drugs. So diagnose, select drugs, switch drug again. This is by trial and error. Sometimes our immune system might come to rescue. Sometimes we'll try with it, we'll end up with only side effects and many kind. So we are with the reactive medical care now. After take the medicine only, like that there are several, the hundreds of the medicine for the diabetes, for the hypertension, and we are trying with them also. Sometimes they do show resistance on the side effect and more. So for that now, because of the genome sequence, genomic variation can be identified, pinpoint the which gene, which pathway they are affecting, lot of studies have been done globally, and the genomic medicine concept has started and treating the patients with the genomic medicines by looking into the information of the patient's genotype and phenotype from 2014. So by looking to the patient genetic data as well as the phenotypic data, they stratify the disease, what kind of disease, is a complex disease or dominant inheritor, recessive inheritor, sex-linked, they can classify, identify the defective genes, the protein or the enzyme, the medications, provide therapy, initiate the preventive measures in the whole family. If it is inheriting in the family, we can tell that, okay, this is the gene is inheriting in family. We can prevent them also by prenatal diagnosis and so on there. So this is the kind of genomic medicine is coming up now, and whether we like it or not, it will be there in any moment. As already people are taking up this, and then I'll give some example next there. So genomic medicine is nothing but the right medicine, right patients, right dose, and right time you have to give them also. One of the disease called familial cholesteremia. The title I may not, but because of the PPT, say familial cholesteremia. Here they diagnose that there's a DNA test, and also they could able to see the mutations in the LDR, the low density lipoprotein, upper lipoprotein B, and the convertase gene there. So they identified variation there. Based on the variation, they started giving the one. Even Craig Venter, he sequenced his own genome. He found that the apolipoprotein is defective in there and started taking the statin 2007 itself, because statin is the one, it's not allowing the clotting of the blood in the arteries, so it is thinning the blood there. So one of the very well studies, the Dutch family, they studied based on the 413 patients and the 1537 the control they used to treat this with a statin as a control, no statin, and the statin for about 15 years they studied. After taking the statin every day, a drug, they about 90% and 85% they had a normal life, whereas without statin, even the 40% there, some of them had a severe heart attack and so on there. So statin became a simple drug to thin the cholesterol level. Similarly, another example, I hope all of you know this lady, Angelina Jolie. Her mother, she had a BRCA mutation, breast cancer mutation. Her mother has the breast and ovarian cancer. Her aunt also has the breast and ovarian cancer. She tested the gene for the breast and ovarian cancer. The same mutation she also carries. So then she undergone the double mastectomy, and she's taking monoclonal antibody to suppress this BRCA gene, not to be overexpressed that, and she is still a model. She is maintaining that genomic medicines, putting her in a normal situations. 2012, the UK government has started England Genomics company, government company. 2018, they completed. They sequenced 1 lakh individuals of the health insurance individuals of the UK. And now 2018, they completed, and most of the cancers, whatever they identified, now they know the pathways and culprit gene. They started treating. For cancer treatment, the UK become the global leader now, because they know 1 lakh samples, and they know what is the pathway, which genes has to be suppressed, which genes has to be activated if the tumor suppressor genes are being mutated and so on. So now the genome medicines, what generally we are going further now is that the screening and the predisposition and monitoring them and preventive measures. So it's a now genomic medicine is going to be a preventive medical care, not the reactive medical care. And whether we like it or not, we are going to take up this, the genomic medicine. Here we are in the genomic medicine era. Human Genome Project, though, is completed 2003, but 2022, some sequences are not able to, the repeated DNA sequences, some sequences are not able to completely sequence. They did the sequencing after 20 years or so, and they identified many gaps, they filled the many gaps and constituted 6.7% or more the analysis of the human genome, which has been done 99% in the beginning, but later they added some of the gaps, whatever is there. And also March 2022, telomere to telomere consortium they established. They also sequenced the first complete sequence of the each autosomes and X chromosome except residual array. They made it. So all the genome sequences, millions of genome sequences, individual genes are available now. All of them have been compiled. The homology has been established. That is called genome sequence GRCH38 or HG19, the version. So all the genome sequences added, the variations are added there. Now we know at what region, which gene, which kind of variation that 38th versions is available now. Even now, maybe another 6 months, the next version will be coming there from this last 6 months, whatever the variations are found in globally, they are going to be add that also. That way, the reference sequences. When I say that we have some gene sequences, we sequence the exomes of the disease, we'll go to compare with the standard reference sequences, then only we say that this is variant or not. May 23, last year, they made a human pan-genome references using different ethnic groups. They compared and they said that now all the ethnic groups, when they compare, 99% is accurate, the genome sequence. They identified 700 haplotype of 350 individuals belong to different ethnic groups, and finally added new variations, new kind of gene duplications and additional structural variations are said. So error rate now with the genome sequence is that 1 base per 2 lakh assembled bases or 200,000 bases there. So error rate is reduced now. So after having said this, I'm very fascinated to tell about our own the Indian Genome Project, which has been announced in this group, the Indian government group, that Department of Biotechnology initiative of this to sequence 10,000 human genome. They sequenced and they announced that February 28 last week. So what is the objective of this? To develop predictive diagnostic indicators for several high priority disease and uncommon genetic disorders. So we need to have our own reference sequences because we are different from the Western data. And this is the one Indian government has started. It's a very, very good news for the genome scientists in India. And we are very eagerly waiting for when they are going to open this database also. They established their database here. The 99 ethnic groups they use, spanning to 20 centers all over CCMB, IAC, IJB, many the good labs, the government funded, CSR funded, DBT funded, DST funded. The labs have been sequenced them, they are added here in this. The first phase is the sequence. That the second phase of the objective of this, the project, the Human Indian Genome Project, is that to identify the very risky disease, cardiovascular diseases, mental illness, and cancer. These are the 3 diseases that they wanted to take the samples from that, identify, compare with our own databases, identify each other culprit gene. So now we are established, the Indian government has established Indian Biological Database, IBDZ. IBDZ, it has been located in Faridabad, Haryana, and it has been inaugurated in 2022, and our own data bank. And because we always use the NCBI, I hope most of you know, and EBI databases. Now we will going to have our own databases, and we are the equivalent to many other the country in genomic science also. So what for this databases is needed? The genome sequence is needed to gain deeper insight into India's population diversity, improve diagnostic methods and medical counseling, find genetic predisposition to disease, develop personalized or customized drug, improve gene therapy, and throw more light on individual susceptibility to infectious diseases. So the whole genome sequencing facilitate the reading of personal individual genome to identify differences from the average human genome. And these differences, if you find the differences, then what you're going to do that? From what you going to get from that? The humans, each human susceptibility genes, future vulnerability to the diseases, their reaction and sensitivity to a particular stimulus, and so on. What are the application of the genome project, Indian genome project here? To evaluate rare disease, 10,000 disease and rare 10,000 already diseases are known now globally, and rare diseases are to identify. Tool for prenatal screening, identify. CRISPR-Cas-based repair system. In public health, we already know that COVID-19 pandemic has been addressed by several thousands of COVID coronavirus sequencing. This can be used for the population level, and where the database is available, we can predict this community, this caste, another 5-10 years that, oh, this community, this is the kind of the pathway is affected, this gene is affected, this medicine can be given, that using artificial intelligence to analyze our data and identify genomic variation, and it have a high potential for treatment of genetic diseases. So with this, I end my talk. My take-home message is that genetics should be taught, practiced, propagated as a foundation of all life science and medical sciences. Though 1900 Mendel rediscovered genetic lab, but is not given that much importance, especially in medical schools. This is fundamental. If you understand the fundamental, and then you can address the human healthcare as well as the other in even agriculture or livestock industry. Genomics should be treated as one of the specialized field like radiology, clinical chemistry, and pathology in hospitals and hospital research. It is already there in Western country, in the different hospitals, they're using that, but we have to do that. Now, many a time I address this issue in medical universities, hostel, wherever I give this kind of talk, they say, oh, a lot of data to study. They have only one unit, one chapter, or one paper to study the genetics, which has been very, very neglected. And whether they like it or not, they are going to be outdated unless we take up this also. So, finally, genetic diseases are preventable, treatable, and manageable. This is my group, all my talented, bright PhD students contributed a lot for thinking, asking different kinds of questions. And some of the data, whatever present, some of them are, most of them are from here onwards. They are involved in that process. And I thank my MSc students, PhD students, funding agencies, and the families and the subjects who have contributed their blood sample for analysis. I want you to read this, the last sentence, the last page here. Last page, the last sentence, the last word of disease will be scripted by genetics, whether you like it or not. If you ignore an obituary to the ignorance. So we need to know our own genetic system, our own inheritance pattern, and this genetics and genomics should be treated in priority. So if you don't do that, then I hope some of the Kannada known people can understand you had to do it. We cannot sit tying our hand there. So I hope people will understand this. Some of, I mean, wherever the person coming from Karnataka, for others is a message that we have to explore. We have to go explore new thinking, new data, new scientific questions, and understand more about that for our betterment. So I'll stop it here. Now I'll take up the questions now. Oh, I exceeded 1 minute. Okay, thank you. So you can ask the questions. We have time right now. Hello, all are there?
Yes, sir.
Yes, sir.
Yes, sir. Doctor, we are there.
Yes, sir, we are there.
They given me 1 hour, 15 minutes. I taken 1 minute extra because, yeah, that's okay. It's not an issue.
Sir, good afternoon, sir. I am Divyendu Shekhar Mohanty from West Bengal State University, Kolkata.
Okay.
I have a question that means, so why some traits are called treated as advanced and some traits are treated as primitive? On what basis the traits, human traits can be treated as advanced or primitive, sir?
Maybe some traits were simple, some traits were complex. Instead of advanced, this way, I would, some traits are coded by single gene. Some traits are coded by many